Hello guys, welcome to my second YouTube video. I'm Muzamil. As you can see, I'll be recording this video on a treadmill. And come on, I cannot be recording every single one of my videos in my room with that already robotic explanation. This is a YouTube video, not a university assignment. So, how about we kick off today's video like this? Do you guys remember in biology class when we finally got to the immune system chapter, which is usually covered near the end of the biology textbook? We were told again and again that the immune system remembers the pathogen that causes a disease. So when the very same pathogen enters our body for the second or any time after, the immune response will be much stronger and faster. Supposedly so fast that we didn't even give them chance to infect our cells. The immune memory theory works for most disease. Let's take chickenpox for example. The varicella zoster virus entered our body for the first time. We were most probably a kid during it. They entered our body and successfully got us sick. Better late than never, our white blood cells started secreting antibodies to kill the virus. For the already infected cell, they will be terminated by another type of white blood cell. See, I'm trying my absolute best here not to mention those antigen presenting cells, MHC1, antigen MHC1 complex, antigen MHC2 complex, T helper cell, B cell that undergoes mitosis, later differentiation to form plasma cell and memory B cell, cytotoxic T cell that undergoes mitosis, later differentiation to form effector T cell and memory T cell. Wanna know why I refuse to mention those? Because I love you guys, and I know for sure you did not click this video to hear those mumbo jumbo nonsense. For now, all you have to understand is this: other than secreting antibodies, our white blood cells will not forget to memorize the virus by their antigens, which are structural proteins found on their surface. So when they enter our body for the second time, we can skip those slow early steps and jump straight to the part where antibodies are actually being produced. For those who have learned already in this chapter, once we contacted the pathogen for the first time, to fight them the second time, we can skip all these steps and go straight to clonal expansion. Do not be confused, I have to mention that this time it's no longer B cells undergo mitosis and later differentiation to form memory B cells and plasma cells. Instead, it's memory B cells undergo mitosis to form more memory B cells and later differentiation to produce some plasma cells. Before answering the question in the title, it will be very helpful if you can differentiate between immune response and direct effect of cells infection. Those sore throat, runny nose, sudden increase in body temperature that you experience when you catch the cold are all immune response from your body. You should be grateful that you have the privilege of experiencing those conditions because it shows that your immune system is functional and is working really hard to eradicate the virus that are infecting your respiratory tract. For direct effect of cells infection, it's usually more severe, like paralysis from polio, because the polio virus are infecting the motor neurons, or internal bleeding from Ebola, as the Ebola virus infecting your epithelial cells of the kidney. For short answer, there are 160 plus strains of rhinovirus and none of them are identical in the antigen molecular structure. For the long answer, rhinovirus store their genetic material in a form of RNA, and RNA virus is notorious for mutating rapidly. The question now is, when do they mutate? How about we just kill two birds in one stone here? I'll be explaining to you about that and how RNA virus infection spreads throughout the body. So let's go. First of all, bear in mind, in this segment, I'll be using singular terms for better understanding. Alright, thank you. So, the virus enters our body, travels through the blood vessel, gets to the body cell, binds to it, and injects their RNA into the cell cytoplasm. The RNA will hijack the cell's machinery to produce its own structural and non-structural proteins. Later, the RNA will be replicated with the help of the RNA copying enzyme, which is of course produced by borrowing the host cell's machinery. The proteins produced will assemble into a protective shell that holds this newly copied RNA. Finally, this newly synthesized RNA virus will exit the cell to infect other cells nearby. This newly synthesized RNA virus is supposed to be the clone of the very same virus that infected the cell. The mutation occurs during the RNA replication, mainly because the RNA copying enzyme doesn't have what we call a proofreading ability like what the DNA copying enzyme does. So there are errors in the newly copied RNA in the new virus. And when this virus infects another cell nearby, hijacks the whole cell machinery to produce the RNA proteins, this collection of proteins, some of them are not identical compared to the proteins found in the first virus. The final result is, when the third virus is synthesized, it's no longer identical in genetic material and physical structure compared to the first virus, making it a new and completely different strain of the same type of virus that we are talking about all this while. To be completely honest, this was where I started to regret in making this horrendous video, because I did not know it was gonna be this, not really complicated I would say, but yeah, quite difficult to understand it if you do not have a textbook with you right now and you watch this video in one go. But yeah, good luck understanding this segment, and please finish watching this video. This is only my second YouTube video. Give your brother a chance. Alright, so the first and the second virus is identical in physical structure but not in RNA, as there are errors during RNA replication of the first virus, thus making the second virus a variant of the first virus. Now, the third virus is identical in RNA compared to the second virus but not in physical structure, as the second virus gives a different instruction for protein synthesis compared to what the first virus RNA gives to it. Lastly, the easiest one, the third virus is not identical in physical structure and RNA compared to the first virus. Okay, come on, not too bad, right? Yeah, if you fail to understand this segment for the first time, just repeat this video again and again and again. I'm pretty sure sooner or later you will understand it. Yeah, good luck. Before I end this video, allow me to clear two misconceptions that you are probably thinking, especially after the last segment. Firstly, after a virus infects a cell, it will not use that opportunity to only produce a single clone of itself. It will produce many clones, normally in the thousands, which also means there's supposed to be thousands of RNA replication in the whole cell. These clones later will exit the cell in bulk. So does this mean that my first, second, third virus concept was absolutely rubbish? No, I had to use that oversimplified concept to help you in differentiating between a virus variant and a strain, and when are they produced. Secondly, about the emergence of a new strain. Yes, I did mention in the last segment that one mutation is enough to produce a new variant, but this doesn't mean 2 or 3 mutations are enough to produce a new strain, as a strain is only produced after an accumulation of continuous mutations to the point of there's a significant change of the virus physical properties that we never ever seen before in this species. For example, a new virus antigen of the same species. Yep, that's supposed to be all. Stay tuned for my next YouTube video that should be released next week, or might as well not do that, because most probably I will just release 4 videos at once for my YouTube debut. So thank you for watching.